mitosis and meiosis. Now, before we start, let's talk about what mitosis and meiosis is. It is basically a way of copying genetic material to produce 2 in the case of mitosis or 4 in the case of meiosis amount of cells. So let's talk about mitosis first. So there are basically a few steps. First is the interphase. In the interphase, the chromosomes, which look like this somewhat, multiply and turn into the traditional X look, like this. In it, this is the centromere, and this is a chromatid. So it basically divides and then turns to double the genetic material, and it's technically called a chromatid instead of a chromosome. And then comes prophase. Let's erase this. Prophase. In prophase, the cell's nuclear membrane dissolves, so it just turns to nothing, and the spindle fibers start to form. Now, not a lot is going in this process. And then comes metaphase. In metaphase, the chromosomes line up in a single file line, and the spindle fibers attaches onto one side of it. Next is the anaphase. Sorry if my handwriting's bad. In the anaphase, the spindle fibers pull back, causing it to the chromosomes to turn into 2 pieces. Now they're still attached. And then there's this whole and then it's telophase. Telophase is when the nuclear membrane starts to form again and the cytoplasm starts to split off. And then the process of it splitting off is called cytokinesis. And then afterwards, you have 2 daughter cells. Now, this is important because in the next one, which is called meiosis, there will be 4. Now, in these 2 daughter cells, that's what they're called, they are having the same amount of chromosomes as needed. It isn't half because in interphase, the 2 chromosomes multiply to create the X shape. Next, we're going to be talking about meiosis. Meiosis, I guess you could say it's kind of the same thing because in anaphase, it's the same, but basically it is the same process but twice. So anaphase, it just creates the chromosomes, it duplicates them, and then in prophase 1, the nuclear membrane dissipates, I guess you could say, and spindle fibers start to form. Now, one difference between metaphase in mitosis and meiosis is the first one, it was a single file line, but in metaphase, phase of meiosis, they're first in a double file line. And then Anaphase One, the chromosome split as earlier. I guess you can't say split because they aren't breaking apart. They're just going into to, let's say there's an animal with 4 chromosomes, 4 pairs, like a fly. Then it duplicates them and then makes them like split apart. So there is like 2. There's still 4, but in a different way, you could sort of say. And then telophase, it just splits apart as normal. Prophase, as earlier, same thing. Metaphase of meiosis, metaphase 2 is different from metaphase 1 because, as in mitosis, it follows a single file line and then splits off. And then after that, it does the normal cytokinesis and telophase, and now it results in 4 daughter cells, each with half the amount of chromosomes, because you split it twice instead of just once. Now, interphase, I guess you could say, is the most important part because that's where the cell gets ready for mitosis/meiosis. The length of interphase varies because some cells, such as skin cells, don't have like a long interphase, they have a short one, but others, such as nerve cells, are always at interphase. And that's all for today. Bye.